Vineet Bafna is a professor of computer science and engineering at University of California, San Diego. His research focuses on computational molecular biology and bioinformatics. He also holds positions at the Morris Cancer Center and the Halicioglu Data Science Institute at UCSD. Vineet, thank you so much for taking the time today.
It's my pleasure. Thank you for having me, Joe.
So to start, I'd love to hear more about your research. Can you tell us about your current research on cancer?
Sure. And before we talk about that, I just want to confirm that I'm actually a computer scientist by training, not a cancer biologist. And we certainly don't perform biological experiments. But what has changed is that, the important molecules in our cell, like DNA, RNA, proteins, they can all be expressed as strings. So you could think of DNA as a long string over the letters A, C, G, and T, and it's sort of packaged into distinct uh, strings that we call chromosomes. The total DNA per cell there's quite a bit of DNA. It's about 6 billion letters. And these chromosomes contain genes, which are regions which have the instructions to make proteins, and then the proteins can be thought of as the machinery that makes cells work, right? So that's sort of the background. But the fact that they are strings allows us to store them and analyze them on computers. And in cancer, as in many other diseases, these DNA can mutate. So the letters might change, and that would, then change the instructions to make proteins, which can impact either the amount of protein that is produced. It can go low or go high, and also the type of the protein that's formed because the instructions themselves have changed. So if you're unlucky, this mutated DNA can lead to uncontrolled, proteins that uh, sort of promote uncontrolled proliferation, cell division. Eventually, that leads to tumor formation. And very broadly, my research is focused on studying these mutations, as looking at patterns in these strings and trying to correlate these patterns with other patterns to understand what their impact might be on tumor formation and so on. So that's how I work for cancer biology.
So just kind of following up on the DNA, and you said that the DNA can mutate the instructions to make protein. Is it the case that we can influence DNA and how it mutates and changes the instructions? Are we able to do that at this point?
That is a very good question, and sort of yes, but just to say that in cancer, these mutations often arise sort of in an unfortunate set of circumstances. Your environment might have some mutagens that might change the DNA, and like smoking, for example, could do that. So that's how it works in cancer. But yes, I mean, your question, which is an aside, is can we manipulate the DNA and change it? And yes, there are modern technologies that are allowing people to change DNA in a way that could be beneficial. In fact, the Breakthrough Award last year went to David Liu. He's one of the people, and not related to my research, of course, but that was on editing DNA. And there are many other similar research efforts. Yes.
So I also wanted to ask you kind of a definition question. What is extrachromosomal DNA formation?
Right. So that's actually very pertinent to my own research. It's a very specific type of a DNA lesion that we mostly see only in cancers. And as the name implies, what happens is that in the cancers, in the tumor genome, pieces break off and they circularize. And so you have these circular structures, and you can see it in my background. I just put that up. You can see these large circular structures in an image of the cell. These are still DNA. They are actually human DNA, but they are independent of the chromosomes, and that allows them to behave like little parasites. They can replicate on their own, and when the cell divides, they break off in unequal amounts in different cells. Now, think of this extrachromosomal DNA as a molecule that contains a gene that promotes cancer formation, for example. It might happen by accident, but if that happens, and a cell gets more copies just by chance because of this unequal segregation, then the cell with more copies is more likely to divide, and that will increase the number of copies of this oncogene over time per cell, and make the cell much more likely to promote tumor. And so they are therefore quite influential in both uh, uh, increasing the amount of, the probability that you have tumor, but also just to increase its pathogenicity. And we didn't discover them. They were actually discovered almost 60 years ago in pediatric cancers. And for a long time, people did work on them, but they were thought of as oddities. But what our research found is that they are actually ubiquitous. Almost a third of all individuals with cancers have them, and almost two-thirds of all the cancer types carry ecDNA, and they play a much more important role in cancer than previously imagined. So just studying them, studying their impact on cancer, and studying the vulnerabilities that will allow us to target them are all very, very important now.
So when you say ecDNA, that's the shorthand for extrachromosomal DNA, is that right? So
Excellent.
is there are all ecDNA associated with cancer? Are there some positive ecDNA, or is everything that we associate with ecDNA also associated with cancer formation?
Right. It's a very good question. And we have looked, oftentimes when we study cancer cells from people, we also, from the same individual, we also have their blood cells and cells from adjacent tissue, which is non-cancerous. And we have almost never, I would say, never found ecDNA in the normal cells. We've only found them in cancer cells. So, to our understanding, they are not found in normal cells. And even though they might be created by accident, cells usually get rid of them very, very quickly. But in a few cases, when they contain these tumor promoting oncogenes, they make the cell more fit relative to other cells. And then in those cells, these ecDNA increase in number, and they also increase the pathogenicity and the likelihood of sort of tumor evolution in those cells. So we see them quite often in cancer and almost never in normal cells.
You've used the term pathogenicity a couple of times. Could you define that for the audience, please?
Right. So what we have found, we are at a very early stage, and we have done some very crude analysis. But what we have found, for example, is that when individual tumors have ecDNA, the survival times are much worse. So the patients do not seem to survive as easily. They are patients which have ecDNA in their cancers. The ecDNA, the tumors evolve very quickly. They quickly become resistant to drugs. They are often not sensitive to new forms of therapy like immunotherapy for cancer and so on. So in almost every way that we could measure them, we find that the worst or the hardest cancers to treat are the ones that contain ecDNA.
Hmm. Uh, so I want to talk to you about how this research is done. Can you talk about kind of the things that you do in the research and how you conduct this research?
Right. So before I answer that, I want to just, reassert that this is very much team science. And about 10 to 12 years ago, I read a paper by a scientist, Paul Michel. He was at UC San Diego at that time, but has since moved to cancer. And it talked about ecDNA, of course, but of a very interesting sort of behavior of ecDNA carrying cells when they are treated with drugs. And a paper appeared in Science. It was very interesting and somewhat complex paper. And we got to talking because I was interested in the rearrangements that you see in cancer DNA. And that was a really wonderful collaboration. And we worked together for many, many years, but also a few years ago, he assembled a very large team of basic scientists and a lot of people, just the experts in many fields, basic scientists, bioengineers, cancer researchers, mathematical modelers, and even computer scientists like me. And our team now is about 60 people with 13 senior people and a host of other sort of junior people in the lab. And we meet sort of every 2 weeks. It's a very, very collaborative effort. We also meet in person a couple of times a year. And yes, people collaborate very, very closely with their expertise, and everybody learns a little bit from everybody. So I would say that in this case, besides the exact work that we do, how we work has also turned out to be very, very productive. Just assembling the team, developing rules where everybody can contribute as an equal, but still are focused on the ecDNA. I think it's a lot of credit goes to Paul to really build this team out. And we are, as much as I'm talking right now, I'm also a representative of that team. Now, my own research, of course, is on the computational part. So what we can think about is what is in the ecDNA, right? So the way we could do this is we can sequence ecDNA, which means that we extract the total DNA from the tumors and send it off to sequencing machines, and they give us short strings that are sampled from this tumor DNA, which contain the DNA of the tumor. And then we compare it against a normal reference, and it tells us what changed, what regions have increased in copy number, what regions are not, far away on the human genome, but because of the circularization have come together on ecDNA. We analyze these patterns. And generally, sort of in terms of methodology, we convert these strings into combinatorial graphs or networks and identify cycles, paths in these graphs. And that sort of help us say what's on the ecDNA, which samples carry ecDNA and things like that. And we have been working on this for many years now. So we have analyzed close to 40,000 cancer samples. So besides sort of working on individual cancers, we also get a sense of how different cancers have different, structures of ecDNA, what genes are more common in a particular tumor type. For example, brain cancers might have amplification of the gene called EGFR on ecDNA, but other cancer types, such as ovarian and breast cancer, might have an amplification of HER2 gene. So different cancers that might both carry ecDNA might still have different properties because they have different genes that are amplified. And the other things we look at are to correlate the presence of these ecDNA with other clinical variables, with other basic science research that other people have done, and to find if there are some genes that are, for example, not on ecDNA, but they are essential to maintaining ecDNA, because if we could stop those genes, if we could target those genes, that might destroy the ecDNA and provide a new therapeutic opportunity. And so we co-founded a company called Boundless Bio. It's a public company now, so I'm not directly involved with it, but it has developed some novel molecules that are in clinical trials, and so the work is quite exciting at this point. Yeah.
So you mentioned these short strings that you look at, and you also mentioned earlier that I think you were saying DNA or there's something that has 6 billion letters. So when you say you're grabbing a short string, does that mean you're getting a portion of the total number of letters that would describe what you're looking at, or are you getting you're getting a view of what the whole thing looks like? How does that happen, and what are you able to see?
Right. It's a good question. Absolutely right that there are about 6 billion letters. And it's really because we get one copy from our mother and one copy from our father. You could think of these 6 billion letters as being two almost identical copies of 3 billion letters each. So that's how we should first think about ecDNA. When we sequence today with some of the technologies, we get little pieces of these letters, about 300 base pairs. So it's a very small fragment relative to 6 billion, but we sample many times from random locations. So on the average, every region, every nucleotide, every letter is covered about 30 times. And so it's a large amount of data, but broken into small pieces, sampled at random from a very, very long region in very, very short pieces. So you have to kind of put them together like a puzzle and figure out, in our case, we have to figure out not only that it's, how it's different, but also some of it is not even on the chromosomes. It's broken away and circularized, and you can see through their overlaps that where the circular structures are. So that's how we piece it together.
So what's stopping us from being able to get all 6 billion and read those at a pretty like granular level?
Great question. So we do actually sample all 6 billion. And one way you could think about it is from those small 300 base pair sequence, you should be able to get the full picture, right? So you could take some 300 base pair sequence, and then you take another one, which is, say, very similar in the ends are very similar. So you can join them, and then you can progressively join them and get the entire 3 billion. Unfortunately, that technique doesn't work because a lot of DNA in our genome is very repetitive. So you might think of a road, and that road has many junctions which lead to other roads. But if there's something identical, same set of houses, completely in a different part of the geography, you could sort of jump from one and you wouldn't know that you're talking on one section of the road or the other section of the road. So they are completely identical on the human genome. And when you sample them, you don't know if you're looking at one part or the other part. And that's why you can't piece them together. But you can sort of compare them to the reference and say roughly where they might be from. And you get an imperfect picture, from which you can derive the properties. And since we are mostly focusing on the part that breaks apart and forms ecDNA, we focus on those, but we also look at the other regions to see if there are changes in those, and there might be some interactions between the ecDNA part and the non-ecDNA part. So all of that is part of our research.
So that leads me to my next question, which is what does technology or how does technology enable your research? And kind of pulling the thread of the last point, is there a way that we could get a perfect picture? You said it's kind of an imperfect picture right now. Is technology allowing us to get a more perfect picture?
Yes. So biotechnologies, there's just been a revolution in the last 10 or 15 years that made this work possible. I should also mention that before I joined UCSD, I worked for a while at a private company called Celera, and that company also sequenced the human genome. It was the first sequence that was the draft sequence that was produced in 2001. And that entire process cost literally hundreds of millions of dollars. So to produce one reference sequence. Today, we can actually routinely resequence a genome for only a few $100s. So there's a huge change in that technology. But of course, I should just clarify that it's not an apples to apples comparison, because previously there was no landmark and we had to do the sequencing, but we also had to assemble it, prepare a high quality reference. And today, when we resequence a tumor genome, we compare against the reference, so that part becomes easier. And so the reads that we are generating, the technologies are very high throughput and very cheap, and we can quickly sequence a lot. But of course, they're not capable of fully assembling into a reference. It's only by comparison to that reference that we can make our inference. But that process has become very cheap and very expensive with that, which is why we are, sequencing tens of thousands and soon hundreds of thousands of tumor genomes, and can do the kind of analysis that wasn't possible 10, 15 years ago. So that's one thing. Uh, we also have lots of new technologies which are, for example, tell us about how DNA and DNA interact or DNA and protein interact. These are so-called epigenetic technologies, and that allow us to add more functional information, not just what's on the ecDNA, but what kind of proteins interact with it, how these, ecDNA regulate or switch different genes on and off. And all that is now possible, which again was not possible some years back. But of course, we could love to see even more improvement in technology. If we could get longer strings, now we have some new technologies that are more expensive, but can generate strings that are tens of thousands or even hundreds of thousands of bases long. And that allows us to clarify these more complex repetitive regions. And there might be a time in the future where we could, the lengths could increase even further, maybe as much as an entire chromosome, although we seem to be far away from that. So that's one place where technology could help. Another place where it could help would be where you could sequence the content of single cells rather than connecting a lot of different cells, collecting DNA from a lot of different cells and sequencing them in bulk. And that will tell us if there's a difference between, from cell to cell in their ecDNA content. And then finally, we could have technologies to isolate just the ecDNA part or separate it at least from the chromosome and only sequence that. So there's a lot that we could look forward to in the near future. And this is just biotechnology. I haven't even mentioned all the different sort of computational technologies that are now available. Computing has become very cheap. You could compute on, different architectures, different GPUs, and so a lot of computing resources are available to us that were not available even, say, 15, 20 years ago.
Yeah. Well, related to computing, how is AI influencing how you're doing this work, how you think about this work, and what potential there is for the work?
Right. Also a good question. And the opportunities for AI are profound, although I have to say that we don't use it as extensively as we could. But I'll give you some examples where we do use it. So one of the things we do is to also look at images of cells at some resolution. What you see in my background is an image like the kind that we analyze, but it's a more high resolution image. But in a typical setting, we will have some just ordinary microscopy images. And what we want to do is look at these images and in cells when the chromosomes are not as compacted, and what's outside of the chromosome and what's on the chromosome is not as easily visible. In that case, we could use AI to really figure out what makes these the parts that are outside of the chromosomes. So what kind of patterns we see in the images that allow us to confirm that these are actually outside of the chromosome and not on it. So we are developing those technologies. Another thing we might want to do is not actually sequence tumors, because sometimes you want to diagnose if a person has ecDNA without even knowing that they have a tumor. And the tumor cells actually leak their DNA into blood. So can we just take DNA in the blood and identify that weak signal that might be indicative of cancer or ecDNA? And those signals are very subtle, but if we have enough data, we could train AI-based algorithms to identify which cells carry ecDNA. So there are many regions, many areas where AI can have a very profound influence. We are only scratching the surface there. There are other problems where AI may not be as useful. For example, if you want to understand the fine structure of ecDNA, what genes are on it, what are the regulatory switches, and so on. I think here still the algorithmic techniques, which work on graphs and strings, those are more useful, and we will continue to develop those algorithmic methodologies as well. So it's a mix of AI and combinatorial algorithms that we use.
Hmm. So what does the future of this research look like? What do you see on the horizon? Are there any breakthroughs on the horizon?
Yes, I mean, absolutely. So ecDNA, like I said, was a niche field for a very, very long time. It's now a very important subfield of cancer. There are many researchers, I would say in the hundreds, if not in the thousands, who are working very actively on it. And there's a really central question that if these ecDNA are so pathogenic, if the individuals that have them, and they're so ubiquitous that so many individuals with cancer have them, then definitely we should be studying them, because those are the worst cancers. Those are the cancers we absolutely want to treat. But at the same time, the fact that we don't see ecDNA in normal cells suggests that they have some kind of a metabolic load on cells. So cells that carry ecDNA, in some sense, they are more fit, they're more likely to proliferate and divide. But on the other hand, they probably also have some vulnerabilities, because you don't see them unless there's some oncogenes and these fitness developing genes. So if we can identify these vulnerabilities, we might find some unique therapies for ecDNA. And I think that's really the central question. Which individuals have ecDNA in their cancers, and what are the kind of vulnerabilities that allow us to find better therapy? So I think a lot of our research is based on that, but there are also many interesting uh, basic science questions. The properties of genes that are on ecDNA are very different from those when they are on chromosomes. These genes are more active. There are different proteins that bind to them. The ecDNA interact among themselves. The ecDNA might interact with chromosomes. They have proteins. So understanding how those systems behave will then allow us to find the vulnerabilities that will allow us to answer the major question. And because this field has just taken off in the last 10 years or so, there's just a lot of work to be done, and there's a lot of excitement going forward.
Awesome. Well, Vineet, thank you so much for taking the time to share your research and your insights, and I hope we can continue the conversation down the road.
Thank you for having me. It was my pleasure.